Welcome back. In this section, we're going to talk about the cells of the nervous tissue and then look at the nervous tissue and see how those cells are arranged and what they do. So, just a quick one slide review. We learned about the neuron. We know that it sends and receives nerve impulses. And we also learned, maybe you don't quite remember, but this only makes up about 20% of the nervous system, which means the majority of the nervous system is something else, something we've never learned about before. And those cells are neuroglial cells. And so after we're done today, come back and look at these different pictures and see if you can figure out what each one is because there are actually 6 different types of neuroglial cells. 4 of them live only in the central nervous system, and 2 of them live only in the peripheral nervous system. Well, the good news is we're not going to learn about all 6 of them in this course. We're going to learn about 3 of the ones that live in the central nervous system and 1 of the ones that lives in the peripheral nervous system. So, in the central nervous system, we will learn about the oligodendrocyte, the astrocyte, and then the little tiny microglial cell. And in the peripheral nervous system, we're going to learn about the Schwann cell. So let's go through these one by one. First, the astrocyte, this blue cell right here, astrocyte. Looks like a star. It's very star-shaped. And if you look at its processes, you will see that on one side, it attaches to neurons. Yes, it's going to attach to multiple different neurons. Each astrocyte has its own little territory that it's responsible for. And then also notice that it has processes that are going to attach to blood vessels. And in doing so, this allows it to bring the nutrition from the blood vessel to the neurons because the neurons actually don't touch the blood vessels. So they can't get any nutrition from the blood vessels directly. And so this is how it provides the metabolic support. Now, astrocytes also do something else, and that is if there is inflammation or any kind of infection or any kind of injury going to neurons or any area of the brain, they will actually form scar tissue to sort of wall off that area of brain injury to protect the rest of the brain from further injury. So it's kind of like, okay, this part of the brain is injured, but the rest of the brain, you're not going to be affected from it. So astrocytes really important. Now, looking at that tiny little microglial cell, that allows it to cruise through. Think of a little thing that can dart through all the little nooks and crannies. It's very mobile. And it's a phagocytic cell, meaning that it can do phagocytosis. It is looking for harmful substances that don't belong there, little pieces of debris, some protein that has changed shape from normal, maybe some dying cells, some damaged cells, some products left over from inflammation, any bacterial or viral remnants, and it's going to phagocytose, get rid of all of that stuff that doesn't belong there. The third type of cell we're going to talk about in the central nervous system is called the oligodendrocytes. So say that word, oligodendrocytes. And this is the cell that's responsible for forming what is called the myelin sheath in the central nervous system. And the oligodendrocyte, as you can see, has multiple extensions, and it can send this extensions to multiple different neurons. Well, it's basically sticks an arm out and finds a neuron and then just wraps it around that neuron. And then a different arm will go out and it'll wrap around the axon of that neuron. And so these extensions are able to wrap around multiple neurons, half a dozen or so. And so it's going to form all of these different extensions from multiple oligodendrocytes will be forming the myelin sheaths on the axons in the central nervous system. So those are the 3 neuroglial cells that we are learning about in the central nervous system. Now, in the peripheral nervous system, we have one neuroglial cell we're going to learn about, and that's the Schwann cell. And that's the cell that's going to be forming the myelin sheath around the axons. But notice it's doing it differently. It's not sending out processes. It doesn't. This cell doesn't have any processes. What's happening is this cell wraps itself around part of an axon. And so we're going to have multiple different Schwann cells wrapping themselves around the axons, and there's going to be little gaps around them. So it's going to take a lot of Schwann cells to make a myelin sheath in the peripheral nervous system. We'll be coming back shortly to talk about this myelin sheath and the whole point of this. So when we look at the nervous tissue in the central nervous system, we have these huge neurons with their cell bodies, there's dendrites, and then their long axons. And we have all these tiny microglial cells. We learned about the astrocytes, the oligodendrocytes, and the microglial cells. And so how does this correlate to the brain and the spinal cord? So this is a cross section of the spinal cord. And in this one, a special stain has been given, which forms this butterfly-shaped area, which you can clearly see here in the middle. But on this section of the brain, which is done this way, so it's like you have a front section and the back section of the brain. This is what the brain looks like without any stains. And so you can see that there's darker tan areas and lighter tan, almost white areas. So this orangey brown area here on the spinal cord correlates to these darker tan areas here on the surface of the brain, and also some areas very deep in the brain. And those parts of the brain that are the darker gray areas are known darker tan areas in real life. For some reason, we call them gray matter. They're not really gray, they're a dark tan, but for some reason we call them gray matter. And so what is this gray matter formed from? Well, it contains the cell bodies of the neurons. Okay, and because it contains the cell bodies, yes, it's also going to contain dendrites. It's also going to contain neuroglial cells. And it's also going to contain some very short non-myelinated axons. In other words, axons that don't have myelin on them. Okay. So that's what the gray matter is. Now, in the brain, as you can see, there is gray matter right on the surface. And this gray matter on the surface is called the cortex. And that's an important word to know. So we have the cortex of the brain, which is always gray matter. We've got some gray matter very deep in the brain, which has other names we're not going to learn about in this course. And in the spinal cord, notice we do not have gray matter on the surface, but our gray matter in the spinal cord is located deep in the areas of the spinal cord with gray matter actually called horns. Now, the lighter areas of the spinal cord and of the brain are called white matter. And so the white matter is going to contain the axons that are myelinated, that are running together. So axons in the central nervous system that travel together are called tracts. So remember, axons that travel together in the peripheral nervous system, those were nerves. So this is the same thing as a nerve, but it's in the central nervous system. So all this white stuff here, those are tracts. Those are groups of axons traveling together. So in your spinal cord, you've got all these groups of axons traveling down, bringing motor messages, and all these groups traveling upward, bringing sensory messages. We have the same thing happening here in the brain, and we also have messages which are going to cross between the right and the left side. Okay, so in the brain, the white matter is deep to the cortex, all of this area, whereas in the spinal cord, the white matter is superficial. to the gray matter. So let's talk about that myelin. What is this myelin stuff? Well, myelin is very lipid rich covering, coating the axons. So if you've ever heard of Crisco, if you're baking, it's like Crisco. If you haven't heard of Crisco, maybe you've heard of other fatty substances like the white fat that's in bacon. You know how that's very white and fatty and greasy? That's like myelin is, okay? So it's very lipid rich, very fatty coating, and it's on the outside of the axon. And that electrically insulated. So if you ever think of like the wiring in your house, you know, they have a little copper wire and that has the plastic on the outside. And, you know, sometimes that plastic is like, you know, green or red or black. You know, like if you're doing electrical work and you're supposed to hook up the black to the black and the green to the green and the red to the red. So you have positive with positive and negative, and then you're grounded. Okay. That's to isolate the charge wires from each other. So this myelin isolates the axon. So if there's a nerve impulse from an axon nearby, it doesn't affect this axon. But as we're going to find out shortly, the big thing that this myelin is going to do is going to change the speed of conduction of the impulse traveling down that axon. Okay. So this myelin formation starts when you're still in your mother's uterus at about 5 months gestation. But most of this is done in your brain by the time you are have your second birthday. So this is why it's crucial for babies in the first 2 years of life that you have them use baby formula or breast milk. Do not put them on skim milk. Do not put them on 1% or 2% milk. If you're giving them milk as a supplement in their diet, it must be full fat milk because they need the fat to make this myelin coating. And so most of that myelin coating is so rapid the first 2 years of your life. It's nearly complete in your brain by the time you are 2 years of age. So if you have this dietary restriction and this fatty restriction the first 2 years of your life, you can never make that up. And that child's brain will be deformed for the rest of its life. Some sensory areas in your life will take a few more years and won't be completed until about the time you start grade school. And then there are a few association areas in your brain that will take until your mid to late 20s to finally finish. But as you can see, this myelin is a thing that's super important when you are a toddler or before you are even a toddler, the first 2 years of life. Okay, so the myelin sheath in the central nervous system is going to be the oligodendrocyte, whether it's in the spinal cord or whether it is in the brain. And in contrast, in the peripheral nervous system, that neuroglial cell is going to be the Schwann cell. And so the Schwann cell forms like a hot dog bun around the axon, and then it just starts wrapping itself around and around and around the axon like a jelly roll. And notice it's pushing the nucleus of that cell all the way to the outside. And so you end up with 10 or 20 rolls of that Schwann cell around that neuron. And we can see this if we do a special stain of a nerve, and you can see all these different axons. That's what the pink is, the light pink. And then the dark pink is a special stain for lipids, showing all the myelin sheaths. And then the dark black is showing the nucleus of the Schwann cells. Okay, so looking at a drawing of one motor neuron in the peripheral nervous system, we know now this is a peripheral nervous system because these are Schwann cells, okay? This cannot be in the central nervous system because these are not oligodendrocyte processes. These are individual Schwann cells. And you can see that there are gaps in the myelin sheaths, and there are going to be gaps in both the central nervous system and the peripheral nervous system. And these gaps in the myelin sheaths are called nodes of Ranvier. Why? Because they were named after some dude named Ranvier. Wonderful French name, Ranvier. Okay. And so here's a photomicrograph showing the axon here in the middle. Here is the Schwann cell, and so there's the little node of Ranvier, and you can see several others on this other picture to the right. So what's the point of this? Well, as you know, the nerve impulse will travel from the cell body originating in this triangular area, going down the axon all the way to the axon terminals. But when it hits a Schwann cell, what's going to happen is that nerve impulse basically will jump to the node of Ranvier, and then it jumps to the next node, and the next node, and the next node, and the next node. And this is called saltatory conduction. And this changes the speed of conduction. Because what happens normally in a naked bare plasma membrane where you have a naked axon without a myelin sheath, when you have the stimulus and you have that nerve impulse, it's going to be slow, and that impulse is going to get slower and slower the longer it travels. And so that's why the axons that don't have myelin tend to be really short. A millimeter, a half a millimeter, or a quarter millimeter. All those little interneurons in our brain that are very short. Okay. Whereas in a myelinated axon, this part here would be very slow if it wasn't myelinated, but it's going to jump, and it only has to travel a very short period before it's going to jump to the next node of Ranvier. And then it's going to jump to the next node of Ranvier. So what does that do to the nerve impulse? Okay, so we say that this nerve impulse was slow, but okay, in reality, it's about 5 miles an hour. So it's faster than you can walk, okay? 2.3 meters per second. Okay, so if we look, it's going to be a third of a second to get all the way from your pinky toe to your brain, and the same thing to get back down there. Okay, but if you're standing on a hot coal, you don't want to be standing on a hot coal for a whole second. You want to be off in a nanosecond. Okay, but with saltatory conduction, instead of going 5 miles an hour, it can then travel 250 miles an hour, or over 100 meters a second, faster than a NASCAR racer. So that myelin sheath is super important. Okay? So if we're asking what the entire thing is called, the entire thing is called a myelin sheath. If we're asking what the cell is called, in the central nervous system, it's called an oligodendrocyte. And in the peripheral nervous system, it's called a Schwann cell. So pay attention to what the question is actually asking when you give the answer. Okay, so what happens when this goes wrong? Well, an example of that would be multiple sclerosis. In multiple sclerosis, this is a chronic autoimmune disorder. Okay, so what does this mean? Autoimmune disorder means it's an immune disorder, so your body is going to be making antibodies which are going to be attacking yourself. Okay, in this case, it's going to be attacking the myelin sheath, specifically in the central nervous system, not the peripheral nervous system, but in the central nervous system. So these immune cells will either the cells directly, like you can come back once we've learned about the immune cells, and you will learn specifically which cells can directly attach the cell, or which cells make the antibodies which can attach and then be attaching. But the net result is the myelin is going to be damaged. And so we will say that that axon becomes demyelinated over time. Over time, that axon loses its myelin. And so that axon's function gets disrupted. And scar tissue can form here because of the inflammatory reaction. And so that axon may become totally non-functional. Okay, so we're just going to stick with the basics. We're not going to learn any more details about types of multiple sclerosis, signs, symptoms, anything else. I just wanted you to have an example of what happens when things go wrong. Okay, so here's the quick thinking. There's quite a few things to do on this one slide. Start with this big picture. What cell? There's two cells in this big picture. What cell parts? What's the term for this type of conduction? Then look at these three other pictures. What other three cells are there? Where are these three cell types? Oh, well, actually, there's 5 cells. Where are, for each of those 5 cells, where would they be found? And what are the functions for each of those 5 cells? We've covered quite a bit. This is one of the harder sections for the nervous system. So thank you for all your hard work. Make sure you understand all of this before you move on to the next section, which is a little bit easier. We're going to be talking about the nerve impulse. See you shortly.